Okay, could somebody confirm you can hear me?
Yeah, we can hear you.
Thank you. All right, before we begin, are there any questions? I think I'll hold off just a little bit because I just heard somebody log in, so maybe people are still trying to log in. Let me share my screen here. Oh, I should turn on the light too. It's getting really dark in here. All right, let me ask again. Are there any questions before we begin? All right, if not, today is October 10th, and in the lab today, we're going to be discussing Lab 4, stains. Any questions? I guess I'll keep that up. Lab 4, staining techniques. We'll discuss the simple stain, the differential stain, and special stains. There is some reading in the textbook you should do before you do the lab. You should read about the size, the shape, and the arrangement of bacterial cells, about 2 pages, 73 to 75. You should read about the bacterial cell wall, pages 80 to 83. You should read about the simple stain on page 65. You should read about differential stains, the gram stain on page 65 to 66. So that's the page, and the acid fast stain on another page 66 to 67. You should also read about special stains. That's the negative staining for a capsule on page 67, as well as page 75 to 77. And then you should read about the endospore stains on page 68 and 92 to 94. And then the flagella stain on page 68 and then page 77 to 78. Any questions about any of that? There is some supplemental material in the lab today. You'll be watching some video clips, and there is a virtual lab for today. The learning objectives for this lab is one, to be able to prepare a bacterial smear for staining. Two, understand the principles and the procedures used when performing a simple stain, a differential stain, and a special staining technique. Three, interpret the results for a simple stain, a differential stain, and special stains. And four, be able to define the following terms: the different stains, what's the coccus, a bacillus, a spirillum, what is peptidoglycan, what are gram-positive cells, what are gram-negative cells, what are acid-fast, what's the acid-fast stain, what is a primary stain, what is a mordant, what is a decolorizer, what is a counter stain, what is a cell capsule, what is an endospore, and what is a flagella. Any questions about any of this material? Because of their extremely small size, bacteria are largely transparent. It's also because they're mostly water. Therefore, it is necessary to stain them before they can be observed under the microscope. And let me state that's for most bacteria or most microbes. There are a few which are colored, meaning they have their own color, but most of them are not. And when preparing a bacterial smear, all you do is you add a drop of sterile water to a clean slide. I don't know if you can see this. Hopefully you can. Oh, good. The lecture file finished. So now my computer will act a little faster. So you have this drop of clean water to the slide, and then you add the bacteria to the slide. Usually what you do is you have a loop. Let me get like a pencil. I don't have a loop here. And you don't fill up the loop entirely with cells. In fact, in this loop, pretend that's a loop there, you would have it so that you barely see the cells. You don't want it even a fourth of the loop or an eighth of the loop. You want it just barely so you can see the cells on the loop. And then you take that loop of bacteria and put it in the drop of water and mix it around. So mix the loop in the water to spread out the cells, and then you spread that water all over the surface of the slide, meaning there will be water all over the slide. There's two reasons for doing that. That's first, to spread out the cells, get them a single layer thick. And you can only do that if you only use a very tiny amount of cells. If you have even an eighth of a loop, you'll have too many cells, and then you'll have cells on top of each other, even when you spread it out all over the slide. And then the second reason for spreading it all over the slide is you want a very thin layer of water so that water can evaporate within a few minutes. You then allow the slide to air dry, and then the organisms are fixed onto the slide by gentle heating. In the lab, we normally have a incinerator, which I don't exactly have here. Let's pretend this bottle of water is an incinerator, and it should be pointed this way with the heat coming out here, but I'm gonna have to go this way because, well, you can't see the bottle of water. Can you even see the bottle of water? Let me stop my share and then I can see. Oh yeah, there it is there. So it should be this way. So the heat's coming out this way, but I can't have the bottle of water. It will be out of the view if I put it on its side. And then you take the last slide and you gently pass it over that where the heat is coming out of the incinerator. And you pass it over 3 times. Another thing you could do is, how can I do this? Now here we use this eraser. That here will be the hole of the thing, and then this will be the outside. You can also touch the glass slide to the outside of the Bechtel incinerator. Don't put it directly where the heat is. And gently heat up the slide. You should hold the slide with your fingers so that if the slide becomes hot, you know you're getting it too hot and to remove the slide immediately. You only want to warm up the slide. So the slide should be just warm to the touch. Any question about any of that? And the purpose of doing that is to heat fix the bacteria cells to the slide so they'll attach to the glass and they will not wash off in a later step, which is in a step after you make the bacterial smear. So the bacteria smear is the first thing you do. And then after you have the bacteria smear, you will stain the cells. So you allow the slide to air dry and then heat fix. And the heat fixing will not allow the cells to wash off during this staining procedure, which follows making the bacterial smear. Let me scroll down here. For some reason, I've lost my mouse. Not my mouse, but the keyboard here. There it is. Now I can see it. So a simple stain is a technique using one stain to stain the bacteria cells. And you'll notice I scratched out the word basic because a simple stain could be an acidic dye or a basic dye. The purpose of doing the simple stain is to get the cell shape and the cellular growth arrangement. And that's all you can do with the simple stain. All the cells will stain the same color. A differential stain is a staining technique that usually uses 2 or more dyes to provide additional information about the cell. such as the Gram stain is a simple stain which uses two different dyes. And then lastly, we'll talk about special stains. They're used to dye a particular bacterial structure, such as the cell capsule, an endospore, or a flagella. And so the special stain shows up a special part of the cell that you're attempting to look for. All right, this slide is showing you the cellular growth arrangement. Cocci can be in singlets, which are not shown here. They can be in diplococci, shown here, two cocci stuck together. They can be in four cocci stuck together, which we call tetrads. They can also be in eight cocci stuck together that we call octets. And then there can be a clump of cells that we call staphylococci. And they can be a row of cocci that we call streptococci. Now, it's important to understand that when you're looking at the cellular growth arrangement, most of the time you will see more than one cellular growth arrangement. And if you name it, you go by the highest growth arrangement that you see. So whenever you have diplococci, some of the cocci will be in diplococci, but you will have singlets also. And that's because the diplococci simply broke apart. In the tetrads, you can have some tetrads, but you can have some diplococci, and you can have some singlets. With staphylococci, you can actually have all of these arrangements. And with streptococci, you can have streptococci, you can have diplococci, and you can have singlets. It's simply that the streptococci is broken, sometimes into diplococci, sometimes into singlets. Any question about any of that? With the bacilli, you have fewer cellular growth arrangements. You have singlets, of course. You have diplobacilli, and you can have streptobacilli. I suppose you could have clumps, but you can have staphylobacilli. We don't usually call it that, but you could have clumps too. Bacilli do not normally form cocci, well, I mean tetrads or octets. Yeah. With the spirilla, that's the S-type shape or the spirillum, the more bends in it. We normally only have singlets. So it's with the spirilla and the spirochetes that we only have singlets. And usually, unless you only have singlets, you will have a pleomorphic cellular growth arrangement, meaning you'll see more than one growth arrangement. And what you do is you name it by the highest growth arrangement you can see. Any question about any of that? All right, this also is showing you a pleomorphic cell. You can have bacilli, you can have cocci, you can have spirilla, and most cells are either one of these three, I should say most bacteria cells are one of these three, or a combination of one of these three, like you could have a cell in between a cocci and a bacilli, and that we call coccobacillus. And there are a few cells which are pleomorphic, where they have more than one cell shape. There's only a few species that do that. Most of them are monomorphic in their shape, but there are some which are pleomorphic, in which case the cells can take on essentially any appearance. Usually the cells that are pleomorphic are cells lacking a cell wall. There are some bacteria that lack a cell wall. must have a cell wall, but there's one family that lacks a cell wall. We'll talk about that later in the term. Any question about any of that? All right, let's talk about the differential stains. There are two that we're going to talk about. The first is the Gram stain, invented by Hans Christian Gram. So you should, when you name it, give it a capital G. He used it to distinguish between a bacteria cell infection and a patient's cells. However, microbiologists started using it and they noticed that it was even more differential than Hans Christian Gram was using it, and it distinguished between uh, two types of cells. The gram-positive cell, which stains purple, and then the gram-negative cell, which stains pink. And the gram-positive stained cells retain the crystal violet iodine complex after the staining process. The gram-negative cells lose the gram-positive, the crystal violet iodine complex. So here we're looking at a gram stain, and you can see, let's blow that up a little. Gram-positive rod there, gram-positive coccus there and there, a gram-positive, gram-negative rod there, a gram-negative bacilli, not spirochete there, and there's a better one right there. There's a good one there too. Shrink that down a little. The cell wall composition determines the gram staining ability of the cells. The gram-positive bacteria is structurally different in its cell wall than the gram-negative. The gram-positive cells have a thick peptidoglycan layer. The gram-negative bacteria have a thin peptidoglycan layer. Here we're seeing the peptidoglycan layer of a gram-positive cell. You can see it's very thick. And if I blow that up, you can actually see some of the different layers there. 1, 2, 3, 4, 5, right there. And there's, I don't know, over 20 there, probably 20 to 100 there. And here we got a thick peptidoglycan layer, but it's thinner, much thinner than there. And this could be, I don't know, 15 to 20 layers of peptidoglycan there. But it's much thinner, the gram-negative cell peptidoglycan, than the gram-positive cell peptidoglycan. Now, the gram-positive cell wall is fundamentally different in that not only does it have a thinner peptidoglycan layer, it also has this lipid layer right here. Let me shrink that down so you see all of it. And that is the outer membrane of the cell wall. So after bacteria stain with the gram stain, we first use the gram crystal violet, then the grams iodine. This forms a crystal violet iodine complex, which is larger than the crystal violet or the iodine alone. You then apply the decolorizing step, which will remove the crystal violet iodine complex from the gram-negative cell wall, but not from the gram-positive cell wall, as long as you do it correctly and apply the decolorizing agent for the right amount of time. The easiest way to do it, like I said before, is to visually observe it and apply the decolorizing step until the crystal violet stops running. Once it stops running, stop adding the decolorizer. That will cause the gram-negative cells to be clear, and then we stain with safranin to get the gram-negative cells to stain pinkish. And there we're seeing gram-negative cells and gram-positive cells. And this is actually called a gram variable because in this slide, smear preparation, this is one species, and some of the cells are staining purple, and some of the cells are staining pink. What do you notice that's different about these two cells here and that cell there, this cell here, and that cell there? What do you notice that's different about them? Come on, there's two things you should be able to distinguish.
Are they connected to each other?
No, they're not connected, those two cells. Is that what you're saying?
No, the ones that are like right next to each other. So
Yeah,
like
these are connected, but I'm not talking about this cell. I'm asking what's different between this cell here, the one on the right, and then this cell here or that cell there. And it's true for this cell here. Take a look at the pointer, the top of the pointer, that cell there. What's the difference between that cell there and that cell there? There is a difference.
uh, there's a positive and a negative.
Yeah, there's one that's pinkish, so it's negative, gram negative, and the dark one is gram positive. What's another difference between besides their color?
The size.
Well, not really. That cell here is about the same size as that cell there. It's true, this one is smaller, but that one isn't. What's different between that cell there and that cell there? All right. I'll tell you, this cell has a very distinct boundary. It's very clear where the cell is and where the cell is not. It's not as clear with that cell where the boundary is, and not as clear as that cell either. In fact, it looks like this cell right there is broken right there. And these cells are probably dead cells why they have a break in the cell and why the cell looks a little ghostly, not entirely there, looks sort of like a ghost. And these cells are viable, and so their cell wall and cell membrane are intact. And these cells probably are non-viable. And that's probably why the cell is broken, why that cell is mostly a ghost. Okay. And when we stain some bacteria, particularly the mycobacterium, some people call them gram variable because they will stain this way. But in all of the cases I've ever seen where students have gotten the gram variable result, I ask them, how old are these cells? And they've always told me, oh, these cells are 2 weeks old or older. And I inform them to do the gram stain correctly. You have to use it on freshly grown cells. If the cells are older than 2 days old, the gram stain may not work. And so when the students were staining the mycobacterium and they got these gram variable results, I told them this cell here was probably dead, and that's why it's staining as gram negative. This cell here is probably viable, and that is why it's staining as gram positive. So even with the mycobacterium, at least with all the mycobacterium I've been able to look at, I've never seen any mycobacterium that, if the stain was done correctly, where the cells were grown for 2 days or less, it never showed gram variable results. Only if the cells were older, like 2 weeks old or older, did I ever see gram variable results in the mycobacterium. The mycobacterium are gram positive, but they do not stain well. They do not readily take up the dye, so they stain lighter. But if the gram stain is done correctly, where the cells are less than 2 days old, or at most 2 days old, they will stain gram positively, at least to every time I've done it, which admittedly I've really only looked at two species. The Mycobacterium have, I don't know, about 10 species in them. Tips to prevent the gram variable staining of bacterium: avoid using old cells. Old cells will have dead cells in them. Dead cells may not stain correctly because their cell wall is decayed. And then prepare thin smears. If you have a thick smear, cells on top of each other, it may be difficult to get the crystal violet grams iodine through all the layers of cells down to the bottom layer. And then with the decolorizing step, if you have multiple layers of gram negative cells on top of each other, it may be difficult to get the decolorizing agent to wash out the bottom cells, wash out all the crystal violet iodine complex from the bottom cells. So prepare thin smears. The smear should ideally be only one cell layer thick. Heat fix the bacteria only after the smear has air dried. If you heat fix the bacteria and there is water in the cells or on the slide, that water will evaporate if you heat fix, and that can put holes in the cell wall, which can make the cell stain incorrectly. And then avoid overheating the cells. Excess heat disrupts the cell walls, and you can always notice the overheated cells because it looks like they've melted. Use fresh staining reagents and do not over decolorize. If you over decolorize, you will remove the crystal violet iodine complex from the gram positive cells. Do not under decolorize as well, because if you under decolorize, you will not remove all of the crystal violet iodine from the gram negative cells. And hopefully I said that correctly. If you over decolorize, you'll remove the crystal violet iodine complex from gram positive cells. If you under decolorize, you may not remove it all from the gram negative cells. All right, any question about any of that? If not, that's it for the gram stain. Let's move to the other differential stain we need to talk about, the acid-fast stain. Some bacteria, namely species of the family Mycobacterium and Nocardia, have a waxy substance called mycolic acid in their cell wall. These bacteria are able to resist taking up the dye. The Mycobacterium resist taking up the gram stain. They do take it up, but they'll stain lighter than a normal cell. The Nocardia, as far as I know, stain fine under the gram stain. They do have mycolic acid in them, but it's not as thick in the Mycobacterium. These two cells also resist the acid alcohol decolorization of the acid fast stain. And so we call them acid fast positive bacteria, or AFB bacteria, AFB positive bacteria. Initially, all cells, when stained by the acid fast stain, take up the primary stain, which is carbol which is a cherry red color. However, you then use the decolorizing step, and it is an acid alcohol. It will remove the carbol fusion from the acid fast negative bacteria, but not from the acid fast positive bacteria. You then stain the cells with a dye, either methylene blue or brilliant green or crystal violet. It's almost always methylene blue or brilliant green as a counter stain to colorize the decolorized cells, meaning the acid-fast negative cells. You want them stained. At the end of the staining process, the acid-fast stains will stain red. The non-acid-fast stains will be blue or green in color, depending on which counterstain was used. So here we have an acid-fast stain. Blow it up a little bit. The reddish or pinkish cells are acid-fast positive bacteria. The greenish or blue-green cells here are acid-fast negative cells. Any question about any of that? All right, let's move on to the simple stain. The first simple stain we're going to take a look at is also called the capsule stain. The glycocalyx is a outer secretion of the cell, secreted by the cell outside the cell wall. And if the glycocalyx is firmly attached to the cell wall, is more than just water, meaning it has structure to it and ingredients in it other than water, and you can see it with the light microscope, we call it a capsule. In this class, we'll mostly talk about capsules because you can see them under the microscope. A glycocalyx, which is not a capsule, you need an electron microscope to see. You cannot see it with the light microscope because it's mostly water. All right, so let's talk about the capsule stain, also called the negative stain, because this capsule stain is simply using one dye to stain the background of the slide and perhaps the cell, but not the cell capsule. Let me come back to that in a minute. So here is a negative stain right here. And you can see it's staining the background of the cell very darkly. It is lightly staining the cell, kind of a grayish color. But there's the capsule, and it's a clear region around the cell. And it's not stained at all. And this capsule stain, it's not a negative stain because it's using two different stains. It's staining the background of the cell and the cell, but not the capsule, which is also clear zone surrounding the bacterial cell after the negative stain. If a cell has a capsule and then you grow them on a petri dish, they usually have a mucoid appearance. Capsules are not easily observed with conventional staining methods, so we frequently will use a negative stain where all you're doing is you're adding India ink or nigrosin to the cells, and then you're looking at the cells in a wet mount where the India ink may be the only portion of the wet mount, or that it could be water in there and there's India ink as well. And so you're not allowing the slide to air dry. You're looking at it directly under the microscope. And that way the capsule is not destroyed. In the gram stain, if you were to apply the gram stain to the cells, the process of the gram stain, especially the water rinsing, will wash away the cell capsule. And so you will not see it in the gram stain. Any question about any of that? As well as the gram stain, the cell capsule would stain the same as the slide, but actually with the gram stain, it's too rough and the capsule will wash away. All right, so like I stated, this is either called the capsule stain or the negative stain. And you can sort of see why it's called the negative stain, and that's because we're only seeing really black and white. We're clear here. And there's the capsule, why it's called the capsule stain. All right, the endospore staining is a stain that stains endospores. Endospores are metabolically inactive, hardy structures that are produced by some bacteria as a defensive strategy to survive an unfavorable environmental condition. Usually, cells that have endospores belong to two genera, the bacillus and the clostridium. I think there are a total of four genera, but one of them only has one species in it, and the other only has a few. Um, so most of them are in the family Bacillus or Clostridium. When the environment becomes unfavorable, the parent cell will start to make a endospore. And then when the environment becomes so unfavorable that the parent cell dies, the endospore can survive the harsh environmental conditions until the environment becomes favorable to the cell growing, and then the endospore will germinate and return to its normal metabolically active parent cell, meaning a vegetative state. Any questions about any of that? All right, this table is showing you some of the pathogenic endospore forming gram-positive bacilli, members of the family Bacillus and members of the family Clostridium. Any question about any of that? You will need this table for answering some of the questions in the lab today. The endospore stain that we're going to talk about is the Schaefer-Fulton endospore stain. It uses malachite green as the primary stain. It initially dyes all cells green. Heat is then applied as a mordant to get the malachite green inside the endospore. The cells are then decolorized with water, which will remove the malachite green from the vegetative cells, meaning the parent cells, but water will not remove the malachite green from the endospores. Safranin is then applied as a counterstain to stain any of the cells which have been decolorized by the water. So the vegetative and parietal cells will stain pink. The endospores will be a dark green. I'm going to blow that up a little bit if I can get it. Here we have an endospore which came out of the parent cell. There is a parent cell staining red. Here is a parent cell staining red. And as you can see, it is generating an endospore inside of the cell. Why it's called an endospore. Any question about any of that? Maybe that's a better one to see because the arrow's not hiding it. And that one too. Remember, an endospore is not a reproductive structure. It is a survival structure. Let's move on to the flagella stain. A flagella is a thread-like appendage or a whip-like structure coming out of the cell that's used in locomotion. If the cell is motile, if a bacteria cell is motile, it has a flagella because that is the only structure bacteria have for locomotion. So if the cell swims or moves about, the cell has a flagella. Flagella are composed of the protein flagellin, and you need to know this not for a quiz, but for the lab. You can classify bacteria on the number of flagella they have. First of all, there can be a cell with no flagella, which isn't shown here. Then you can have a monotrichous flagella where there's one flagella. You can have a lophotrichous flagella where there's multiple flagella on one end of the cell. You can have an amphitrichous flagella where the flagella are on both ends of the cell. And you can have a peritrichous flagella where there are flagella coming all out of the cell in all positions. Because flagella are too thin to be seen by standard staining techniques, special stains are required to view them under the light microscope. Oh, well, let me return to the flagella classification, and that is when a cell is growing, first it will have no flagella. And then if it is a cell that will become peritrichous, first it'll become no flagella to monotrichous, meaning it'll put out a single flagella. And then it will become lophotrichous, putting out multiple flagella on one end. And then will become amphitrichous, putting out flagella on both ends. And then lastly, the cell will become peritrichous, assuming that it can become peritrichous, putting out flagella all over the cell. My point to stating this is when you see the flagella arrangement, you will see all of them up to the highest order of flagella, meaning if the cell is peritrichous, you can find cells in all of the different flagella arrangements, including no flagella. Because a recently divided cell will have no flagella. It needs to put it out. Any question about any of that? And then first it'll put out one, then several on one end, several on both ends, and then lastly peritrichous. However, if the cell, the highest it can get, is amphitrichous, you will see all of the flagella up to amphitrichous. You will not see peritrichous, and etc. Any question about any of that? You need to know that for the lab. You do not need to know it for a quiz. So here we're looking at a cell with a flagella stain. If I look hard everywhere, I could find one of each example. I think this has two on one end, one there and one there. That looks like it has none. This one has several, at least on one end. And then these are peritrichous here and here and there. Any question about any of that? And what you call this is peritrichous because it's the highest order flagella arrangement in the slide. And the others will become peritrichous if they're given enough time. Any question about any of that? That one actually looks like it's peritrichous, but just much less than that one. All right, know the terms of this lab? You can ignore the references. In this lab, you're not going to be physically performing any of these activities because we are an online lab. But you must understand how to prepare bacterial smears for staining. And a bacterial smear is something you do before you begin the staining procedure. And then you should understand the principles and the procedures of the various staining techniques and be able to interpret those results using a microscope. All right, any questions about any of that? Do not answer the questions in the lab module because the questions have changed. And if you turn it in, I'll warn you and then give you a penalty because I don't want people turning in the lab module. It's too big. If everyone did, my web page would become full and then you guys wouldn't be able to submit further assignments. Oh, I'm not here. Where am I? Guess I can close that down. But let's go to the worksheet. So view the following video clips on how to prepare a bacterial smear and then how to heat fix the bacteria on the slide. And then this is the steps for making a bacterial smear. I've already gone over it, so I won't go over it again. But you should answer the question, Procedure 1, Question 3. What are the 4 common errors when preparing bacterial smears for staining? So you have to list 4 different things there. And generally in the worksheet, I've got all the questions with a question mark or maybe with a colon if it's not a question, but it should be a question. And then go to Procedure 2, view the following clip on a simple stain where you're adding a basic dye. Wash it off, shake off the water, blot it dry, and below are 2 smears using the oil objective lens that were stained with a simple stain. Then answer question Procedure 2A. What question is that? Is that 3A? 3A. What is the cellular morphology seen of E. coli? So give me the shape, not the cellular growth arrangement. And I'll warn you, E. coli is not a spirillum. And then give me the shape, not the cellular growth arrangement, of Staphylococcus aureus, shown on the right. And then 3B, give me what is the total magnification of the lenses used, which is always the same lens. All right, Procedure 3, the gram stain. View the following clip of the gram stain and then practice performing the gram stain using the virtual lab. Just click this link and the virtual lab should start. It may take a moment to get started on your system. Continue.
Hi, Lab Assistant. Welcome.
I'm
All
not going to go
yogurt has
through all of the discussion
Unfortunately,
here.
this
Let
is
me
what. Now take a look at
Get to
this
the part
yogurt.
of the
Look,
gram
let's
stain.
collect yogurt. In the gram stain, bacteria that stain, bacteria that are. We are going to. All right, let's prepare our slide. To prevent bacteria from contaminating our work area, we have wiped down the table with a 70% ethanol solution. We will also light a Bunsen burner to sterilize the inoculating loops we'll use later and help maintain a sterile working environment. You're going to use a striker to light the Bunsen burner.
What you do
The
here,
gas is turned on low already, so bring the striker to the Bunsen burner with the flint side facing down and squeeze the striker to create sparks.
what you do is you just take your mouse and take the object that the arrow is starting with and bring it down and then it'll
We
work.
need to dilute the yogurt before we put it on the microscope. Start by placing 1 milliliter of the diluent into a This is an inoculation loop, a small loop of wire used to collect samples. We'll use it to place a small amount of yogurt into the test tube with the diluent. Start by sterilizing the inoculation loop in the flame of the Bunsen burner.
Should turn
Hold it.
red hot.
After it has cooled down, use the loop to obtain a small sample of yogurt from the suspect container.
Notice
To
it's
get
not
a
red
good
hot
sample,
anymore.
reach deep into the container or thoroughly mix the yogurt first. Place the yogurt sample into the previously measured diluent, PBS.
So we're diluting it because we'll have too much bacteria on the loop.
Now, gently shake the test tube for 5. Use the inoculation loop to transfer a small amount of sample. Sterilize the inoculating loop again in the flame of the Bunsen burner. Hold it. transfer a small amount of sample from the test tube to your prepared slide. Place 2 loops full from the sample onto the slide.
Just follow the arrow.
Tilt the slide to spread the drop out slightly, about the size of a penny. Now we'll heat fix the slide. This prevents the bacteria from being washed away when we begin the staining process.
You would need to air dry this out first. It's a slight mistake in this video. It's showing the drop of water. You don't want to heat fix a drop of water because the water will boil.
Place the slide in a clasp, such as a clothespin, and pass it over the flame of the Bunsen burner. Now we begin the staining process. After the staining, we use 4 chemicals to stain a slide. We apply them in a specific order and rinse the slide off under slow running water in between each chemical. The first is crystal violet. This is our purple stain, which will indicate gram-positive bacteria. Using the dropper, squeeze out enough drops to cover your sample. Let that sit for 60 seconds. Now rinse it under slow running water. Now use the bottle of grams iodine. Squeeze several drops onto the sample, enough to cover it. Let that sit for 60 seconds. Now, rinse it under slow running water.
Okay, that's how you do this.
Now we are going to
MicroLab.
use the alcohol solution. This will wash off the excess dye, leaving only the dye fixed to the bacteria.
You'll note that their decolorizing agent is only 95% alcohol. This means they will have to decolorize for a longer time than 5 seconds because we were using alcohol acetone or acetone alcohol. This is only pure alcohol, and you need to apply it for a longer period of time. I'm just going to word it that way. And I'm going to stop the virtual lab now, but that's how you work the virtual lab. Any questions about any of that? Oops, I didn't mean to do that. Stop the share. I didn't mean to do that. Let me redo the share. I meant to stop the virtual lab. Close that down. There we go. Back to the gram stain of procedure 3. So go through the gram stain. I've got the steps listed here. And showing you the application of crystal violet, all the cells stained bluish purple. We then apply the iodine, and here you can see the cells stain a purple color, slightly different than that one. And then we do the alcohol wash, where the gram-negative cells become clear, the gram-positive cells remain a purple color, and then we apply the safranin, and then the gram-negative cells stain pink, but the gram-positive cells are purple color. So answer the questions in the table, complete the following table. The gram reaction, gram-positive or gram-negative of E. coli, listed here, and give the cellular shape, the morphology. Coccus or bacillus, give it here. For Staphylococcus aureus, give me the gram-positive or gram-negative, and the cellular morphology, the shape coccus or bacillus. Put it in the table there. And here's the picture of E. coli with the gram stain, and Staphylococcus aureus with the gram stain. Any question about any of that? So here, you do need to fill in the table, so it's not a question, it's a colon. And then answer these questions. For each of the following organisms, what is the color you would see? Pink, purple, or colorless after the step in the gram stain. Staphylococcus aureus, after the primary stain is added, what color is it? Put it there. E. coli, after the primary stain is added, B, put it there. C, Staphylococcus aureus, after the alcohol acetone step, what color is it? D, E. coli, after the alcohol acetone step, what color is it? Put it here. E, Staphylococcus aureus, after the counter stain is added, what color is it? Put it here. And then F, E. coli, after the counter stain is added, what color is it? Any question about any of that? For procedure 4, we're looking at the acid-fast bacteria stain. We have a video clip of the acid-fast stain right there. Review the steps of the modified Kenyan acid-fast bacteria stain here. You stain the cells first with carbol fusion. All the cells will stain reddish, pinkish. Then you do the acid alcohol wash, the decolorizer, where the acid fast negative cells will become clear, but the acid fast positive bacteria will remain reddish. You then apply the brilliant green as your counter stain, and the acid fast negative cells will stain green. The acid fast positive cells will stain reddish or pinkish. So this lists the steps of the acid fast stain. Seen below are two smears viewed at 1000x objective lens that were acid fast bacteria stain. Are the organisms denoted by arrows acid fast bacteria positive or acid fast bacteria negative? And then you have to state either with the arrow right here or right there, here and here, or you can do either above the picture or below the picture, state the first picture on the left, state what it was, and then the second picture on the left, state what it was, then the first arrow on the second picture, state what it is, and then the second arrow on the, or the right arrow on the second picture, state what it is. You can either write that here, or you can write it here by the arrow, or you can put it above the picture. You do need to answer this question though. So each arrow must be labeled, or you will get an error. Or stated here. All right, procedure 5 is looking at special stains, where we've got a video clip for the endospore stain and a video clip for the negative stain. The negative stain here is looking at the capsule. Below were 3 bacterial smears that have been observed with the 1000x objective lens. No, excuse me, 1000x total magnification. And that will be figure A, figure B, figure C. A goes to figure A, B to figure B, C, shown here, goes to figure C. Any question about that? So A is telling you this is the Schaefer-Fulton stain, which uses which dye as the primary stain, so name it, and ultimately dyes which cells which are produced by some bacteria to aid in survival. You call it which cell structure as well. What are some examples of pathogenic endospore-producing bacteria? Some examples means more than one. So if you only give me one example, you'll get it partially wrong. You must have at least 2 examples of a pathogenic endospore-producing bacteria. And you can find that in a table in the lab module, if you don't remember. because the lab module did discuss it. And then that's it. So let's move on to figure B. This is which stain that coats long whip-like appendages which aid in motility. So what stain is this? And then state the flagella arrangement type of this picture. And what are some examples of clinically significant bacteria that produce flagella? You can find that in the lab module. And then for figure C, this is which stain that uses India ink or nigrosin. So that goes to here. India ink or nigrosin, which and can be used to detect the presence of a cell structure, which will be, so that would be C1, C2, which is located exterior to the cell wall and prevents phagocytosis. So list the cell structure there. And then C3, what are some pathogenic microorganisms that produce a capsule? And once again, you must list at least 2 different pathogenic microorganisms because it says some microorganisms, and that is plural. Any questions about that? Once you're done with that, do the case studies. There's 2 of them. Case study 1, a 45-year-old female is experiencing respiratory issues. Based on her clinical presentation, the doctor orders a chest x-ray and also does sputum for a acid-fast bacteria culture. So there's the chest x-ray. And if you don't know, this chest x-ray looks odd. It should be totally black, sort of like this region here, kind of like this region here. And this region there is very white. This region here, this region here, this region here, and that region there. And so something's wrong with the lungs, I'll tell you that. In addition to the culture, an AFB smear is made. So there's the sputum, they're making a bacterial smear there, and then they've stained it with the Zeal-Nielsen stain. And given the information below, what is the most probable diagnosis? You must name the species which is causing the infection. Remember, a scientific name is a genus name and the species specific name. So give me the species name. Okay, any question about that? If you give me the disease, I will mark it partly wrong because you're not answering the question. Case study 2, a 64-year-old diabetic patient is admitted to the hospital with soft tissue necrosis. A routine culture and gram stain is ordered. The bacteria grew aerobically. Based on the findings below, what is the most probable diagnosis? So here we have tissue necrosis, and that looks really bad. Discoloring of the foot there and there. And then they grew the bacteria and did the gram stain. So there it is there. And then they did the Schaefer-Fulton stain. And you should get some information from this, both of these slides. And name the species which is causing the infection. Any question about any of that? All right. If you don't name the species, well, this one will probably be wrong, but you could get partial credit if you happen to name the correct disease. But to get full credit, you must give me the name of the species causing the infection, and you can find the name in the lab module. Any question about any of that? Any questions about anything? All right, if not, I'll be here until 8 o'clock to help you answer the questions in the lab or for any other assistance you need after 8 o'clock, or the last student logs out, I will log out as well.